Hello, and welcome to Virtual Investor Conferences. My name is Matt Lateplo, and on behalf of OTC Markets, as well as our co-host, Zach's Small Cap Research, we're very pleased you have joined us for our quarterly Life Sciences Investor Forum. The first live presentation is from MRVL Biosciences Corporation. Please note you can submit questions for the presenter in the box to the left of the slides. You can also view a company's availability for one-on-one meetings through the Schedule Meetings tab found on the conference platform. At this point, I'm very pleased to welcome Mark Williams, Chief Science Officer, Director of MRVL Biosciences Corporation, which trades on the OTCQB under the symbol MBCOF and on the TSXV under the symbol MRVL. Welcome, Mark.
Well, thanks very much, and good morning, everyone. I appreciate the time today. Hopefully you will be intrigued by the story we have here developing at MRVL. Long story short, we think we have a very interesting treatment for the autism disease, and it appears to reverse the social deficits that you see in autism essentially immediately upon dosing of a drug, which I'm really looking forward to showing you going forward. So in deference to our American colleagues, we're Canadian up here, but we still plead the fifth, and we sort of let ourselves be covered by our disclaimer here. As you know, CNS is probably one of the last major areas of drug discovery that we have to really deal with. We've made a lot of very good advances in oncology and obesity, but we still are really troubled with CNS-related diseases such as autism, depression, and Alzheimer's, which essentially covers the full gamut of life from essentially young children all the way to our elderly. And we think we have a compound, MB204, which can address each of these particular CNS disorders. If you are not aware, and I certainly wasn't, autism is a significant problem. About 1 in 36 children, according to the CDC, is affected by this disease. The sort of estimated direct or social cost in the U.S. in 2022 was $350 billion. And what's important to note is that the depression rates in autistic children are about 4 times as high as they are in their neurotypical peers. In the case of depression, mostly affects adults, and 1 in 8 is currently on some sort of antidepressant. Again, loss of productivity due to depression of roughly $326 billion in the U.S. And if you've been following the scientific literature, there are some questions being thrown at the selective serotonin reuptake inhibitors, which are the Prozacs of the world and so forth. There's some questions about the true efficacy. We do know that there are significant problems. It takes about a month for the drugs to work. About 30% of people develop apathy or loss of motivation. So they're not without some negative side effects going forward. And in the case of Alzheimer's, I mean, this is truly a tsunami that's going to face our aging population going forward. We're going to need armies of caregivers at the rate we're going. 1 in 10 over 65 is affected, and it gets worse as you get older, 1 in 3. Again, $320 billion, and up to 40% of those patients have depression. And we're going to focus mostly on the autism today because that's where the strength of our data mostly lies, and there's other clinical reasons to focus on that population. So we have been developing a drug based on a known approved drug called istradefylline. It was approved for Parkinson's disease in 2014 in Japan and 2019 in the US. It has a very, very well-defined target and mechanism of action. It essentially affects dopamine. And this is, as it turns out, probably the reason why it's affecting social interactivity. Dopamine, as you know, is considered the love chemical. It results in bonding between two individuals, and it looks like it may have a significant play in social behavior and autism. It's actually also known, the receptor of the istradefylline is the A2A, adenosine A2A receptor, which, as it turns out, is also the receptor for caffeine. So you can think of istradefylline essentially as a turbocharged form of caffeine. But there's good literature to say that it's effective in depression, autism, and Alzheimer's disease. So it's a well-defined mechanism of action. What we have done is essentially done a very simple but logical chemical modification to istradefylline. We have fluorinated the methoxy ether functionalities. We've got composition of matter patents so that we've got probably the trump card when it comes to dealing with the molecule. And we did this specifically because we thought it would improve the oral bioavailability and decrease the rate of clearance from the body, because the liver is a major source of clearance. And in fact, what we show here very quickly is that we basically hit the adenosine A2A target almost equivalently to istradefylline. We have a 67 versus 75 nanomolar ratio with respect to inhibiting the target. So that's very close. We also, looks like we don't have the same effect on locomotive activity as istradefylline. Istradefylline, as you know, is, as I mentioned, is involved in that's in control of locomotive activity. So at high doses, you kind of trigger non-specific motions and activity, which is not necessarily a good thing. But we see that we can dose to higher levels with our drug before we see these non-specific locomotive effects occur. And then finally, we see that we do improve the oral bioavailability. On the third panel on the right, we enhance absorption. We have, not only is the drug absorbed to higher levels, but it also lasts in the body longer. So we would think that we actually have a superior form of istradefylline. And we're not developing it for Parkinson's, just to be clear. We're developing it for autism, depression, and Alzheimer's. So we think our adenosine A2A antagonist could be actually a first-in-class treatment for autism. And again, I mentioned the autism before. It's a broad spectrum. It's called, it's considered autism spectrum disorder. So there's a continuum with respect to the disease, but there are two orphan diseases, Rett syndrome and Fragile X, which have highly defined genes associated. They're called monogenic diseases because they're associated with one gene. So they're a very targeted clinical population, and we know what that gene is. In the case of Rett syndrome, approximately 1 in 10,000 females has it. It's a specific mutation in what's called the MECP2 gene, and there is currently an approved drug called trofinetide, and I will show you some head-to-head studies of our drug MB204 against trofinetide coming up. Fragile X, it affects essentially 1 in 7,000 males or 1 in 10,000 females. Again, the FMR1 gene is affected by this disease. There are no approved drugs for this disease at this point. So again, specifically with respect to Rett syndrome, trofinetide is the approved drug. It was approved in 2023. U.S. sales in 2020, or U.S.-based sales in 2023, I think were 177 million. The drug was acquired by an Australian company, by Acadia, and sales are estimated to be in once full market penetration and worldwide regulatory approval, probably exceed $1 billion a year. The cost is $325,000 to $950,000 per year per patient. Surprisingly, you have to take an enormous amount of drug, up to 25 grams a day, which is a lot of drug. If you're thinking of a Tylenol, that would be the equivalent of taking perhaps like 50 Tylenols in order to treat your disease. It's that kind of level of scale. On Fragile X, there is a drug called zatilimast in phase 3 trials, although it's not approved yet. And for the most part, it's just symptomatic relief for these diseases. The trials, what are attractive about these diseases is that the trials are comparatively small, certainly smaller than you would have to go for, say, an Alzheimer's disease. You can see the number of patients is sub 200 into the largest studies. Dosages or time of treatment is as short as 28 days to see effects and as long as 56 weeks. So these are very manageable for small companies such as Marvel. Although we recognize that Alzheimer's is a large disease. And so we are very empathic to the fact that, if you have a drug that can go after both of these, we might be a very attractive takeover target for a pharma. Certainly Acadia, Pharmaceuticals has an interest in Alzheimer's as well as Rett syndrome, for example. Just to go over there are some good deal flow occurring in this space. There was both the Rett syndrome and the Fragile X have had diseases done where there was reasonable deals completed and consummated fairly early on in clinical development. In the case of the Fragile X, Shionogi purchased the private company Tetra Therapeutics with a biobucks deal of up to $500 million, which was something that we can model future deals on and so forth. And again, Neuron did a deal with Acadia Pharmaceuticals, and you can read the terms out there. And feel free to contact us if you'd like to get a copy of the presentation and learn more about the deal flow and our intents to partner our compound. So moving to the immediate effects. So what is remarkable is that prior to showing you the data, it appears that MB204 essentially has an immediate effect on restoring social interactivity. This is, I will go through it. This is a single dose study. Animals or mice that have the same gene associated with an autism condition. This is called an OPRM1 mouse that I'm going to show you. Within an hour of dosing, we can see virtually a total reversal of the social deficits that you see in mice associated with the autism condition. Before I show you that, I also want to show you that totally independent of the autism, we have a very profound and also immediate effect on depression. Remember, I mentioned in the serotonin reuptake inhibitors like Prozac often take a month to show a signal. And that is not the case with MB204 and the target, the A2A receptor. After a single dose, an hour later, we can see a substantial and dose-dependent reduction in the symptoms associated with depression. And it turns out, as I mentioned, depression is a significant comorbidity associated with autism and also Alzheimer's. So here's a totally independent study showing an immediate effect on depression, which is associated with this. And this had no effect on nonspecific movement. So the animals weren't moving around more or anything like that, which can be a confounding variable. So this is, again, giving us some confidence that this molecule is quite a potent molecule and has an acute effect. So I don't want to overwhelm you with data or anything. That's not the purpose of the talk, but it is to hopefully give you some strength and confidence that what we're seeing here is very potent. And again, just to go over this, within an hour of dosing our compound, we're testing social behavior in mice. And by social behavior, you monitor things like personal interaction, touching. For example, the mice touch noses or they touch paws, for example. And that's a surrogate model of social interactivity. And what you're seeing here is, and this will be true of essentially all of our studies going forward, on the first right-hand side in the white bar, or the left-hand side rather, you can see a normal animal given vehicle, a placebo. Then you have a normal animal given a low dose of our drug, 1 milligram per kilogram, that's the light gray bar, and a normal animal given a high dose of our drug, which is 2.5 milligrams per kilogram, which is not very much drug, by the way, certainly nowhere near what you give trofinetide, which I'll show you in a second. The fourth bar, that light pink, is an autistic animal. And what you're seeing here in that first panel, nose contact, is that there's clearly a significant reduction in the nose contacts that the autistic animals make overall. But then there's this, in that light pink bar, that's a low dose of our drug given to the autistic animal. This is an OPRM1 mouse model. So this is not a Rett syndrome or a Fragile X. It's a third model of autism. But you can see this restoration of nose contacts after 1 hour of dosing the compound. And the stars designate statistical significance. So 1 star is good, 2 stars very good, 3 stars great, 4 stars are excellent. So you can see here that on most endpoints, we're seeing 4-star reversal, which is about as much as you want to put down on a chart, by the way. It really doesn't go beyond 4 stars when it comes to reporting statistics in biological systems. But what you can see here, so yes, in the first case, we are essentially restoring to normal levels animals with autism to their normal peers. So the second panel, the middle panel there, this is the time they touch noses. You can see that's also low in that fourth bar, that light pink bar. That's the autistic animals. And then bang, we restore that to normal times. The third panel is the duration of the nose contacts, how long they stay touching. And again, we see a complete restoration. And just to point out to you, because this will become relevant when we do clinical studies, it's already been shown in healthy people given a single dose of istradefylline that they appear to view social situations more favorably. So the same drug that we base ours off has shown clinically to have an effect on social interactivity endpoints. So again, there seems to be clinical evidence which backs up this acute and immediate effect when it comes to social interactivity with our drug. We did this, by the way, in collaboration with a group in France, 2 researchers at the iBrain Institute in Tours, France. So this is, again, third-party data that we're showing you here. I'm not going to show you all the data here, but now we're talking about paw contacts. And this is where it gets actually very intriguing. And if you look very closely, you can see here in this case, the autistic animals, the fourth panel there on the fourth bar on the left hand panel, they don't touch at all. But then bang, we completely reverse this with a single treatment of our compound. And in fact, if you look at that dark purple and compare it to the white bar, it looks like our autistic animals are behaving more sociably than normal animals. And they're doing it statistically significant. This is a remarkable finding. I mean, I don't want, like, imagine the gravity of that. If you take a drug and essentially within an hour of taking the drug, you have this immediate reversal of social deficits. I mean, if that translates clinically, I can't imagine what that would mean to parents to be able to have a social interaction. There would be that there could be hugging, for example, eye contact, things like that, that might be restored. So this is very exciting data, I believe. So yes, this is us now showing the duration of paw contact, or the time that they touch paws. Again, this is the middle panel. You're seeing the same kind of phenomena occur. They don't touch paws in the autistic group. We show a nice, elegant dose response with our compound. And in the case of the higher dose, the 2.5 milligram, which is still a very low dose when it comes to human dosing, by the way, we're actually seeing an effect above and beyond the normal levels. And then finally, the duration of paw contact. Well, the autistic animals don't touch paws at all. So we're totally restoring that to normal levels after a single treatment. All right. Also, in this case, we're showing grooming. As you know, self-grooming is a behavior you'd rather not see in mice or potentially patients. And we're able to show that we reduce grooming as well, which is a very important and very nice benefit of the drug. And suffice to say, again, I point out to you the number of stars. Essentially, on every single endpoint that we're studying here, we're seeing a reversal of the social deficits. There's also 2 other endpoints here. Following episodes, mice follow each other. That's an indication of social interactivity. We restore that. That's going to be important in the next experiment that I'm going to show you when we dose our compound chronically. And finally, there's an immediate effect, dose responses, and every endpoint improved. So with that, that's really exciting. So we actually moved forward and wanted to see, well, what happens if we dose animals for a longer period? So in this case, we did go and use the Rett syndrome model. So this is where the trofinetide drug was approved. It's called DAYBUE. But let me point out to you, you cannot dose in mice the human equivalent dose orally. We actually had to inject trofinetide in this case. So we're injecting trofinetide. We're giving them 100 milligrams per kilogram. And in this case, with MB204, we're giving 10 milligrams per kilogram orally. So a very important distinction here. Now, what you're seeing here is very similar to the experiment. The big one to look for is these 2 middle panels where you have these MECP2 knockouts, they're called, in the blue. The one is an autistic animal, and the dark blue, the light blue bar that you see here that's full is the trofinetide treated Rett syndrome animals. And on the far right-hand side, you can see autistic animals, and the purple bar is the animals given 204. So that's really where you want to focus your attention. And what we've done here is to make it really easy to follow is every place you see a green check mark means we are outperforming trofinetide. So on the number of nose contacts, you can see here that we are outperforming. The time we spend contacting nose, that's the bar below that, we're affecting. Apologies, you can't see the title for that. The paw contacts in the middle panel, again, the purple bar, you can see that we're restoring it. The trofinetide, the blue bar, did not do anything. There was no effect. The time spent touching noses, well, clearly, if you're not touching a nose, there's no time. But you can see, and certainly in the case of 204, we're restoring it to almost normal levels again. Grooming and following. Grooming, we're not having, we're essentially, there's almost no effect. These animals don't groom very much at all. But certainly in the case of following, you can see the trofinetide animals in the light blue do not follow very much, whereas they do in the 204 group. So on almost every single measurement, we are outperforming the approved drug trofinetide right now. Now, what we did is that's an immediate effect. We then stopped the drug and see what happens a week later. And you can see that's called a carryover effect. And you can see after we stop treatment for a week, we're still seeing the nose contacts on the left-hand side are still very high. The time that they're spending is still very high. The paw contacts, the middle panel with the green check mark, we're still touching paws, and they're still touching paws for a long time. Again, almost normal levels. The grooming, there's very little effect on grooming. They didn't really groom too much to begin with, but that's still lower and almost to control levels. And then the following, we're still seeing animals treated with 204. The mice are following each other, which is an indication of their then we now we release animals for 14 days, and we're still seeing an effect. And there's no effect being seen with trofinetide here. So in the case of nose contacts, that's still seen. We're still seeing some residual effects of nose contact. Now the effect on paw contacts has now disappeared after 14 days, where the drug has been washed out of the animal. But in the case of following, the animals are still following each other, so they're still very social, okay? And then finally, when there's this test called the social preference test, where you give the mouse the choice to interact with either a toy or a real mouse, and the more time they spend with the real mouse is quality time. Yeah, I guess it's quality time with adults. You can see that after 7 or after 14 days, while their animals are being treated, trofinetide does have an effect, as does 204. So check mark to trofinetide. But after 10 days of being released, the drug is no longer active in trofinetide, but it's still active in the case of our MB204. So we have a significant carryover effect here. We also have a collaboration with another group called FRAXA. It's the Fragile X Philanthropy Organization. They are independently testing our molecule for us. We're expecting that testing to be started shortly, and data should read out in Q1 of next year. But istradefylline has already been shown to be active in this model, so we're very confident that this molecule should work in this model. So that is an example. There was a question of do we have academic or strategic partners? We certainly have academic partners. All of our research is conducted by third parties. We do not run our lab. We contract all our studies to third parties. Okay, I'm not going to spend too much time on here, but really I want to end on saying that there is blue sky associated with MB204 and our company, and that it is well known actually that if you take coffee, it protects against Alzheimer's. And if you remember early on in the conversation here, I mentioned that istradefylline is essentially a turbocharged form of coffee. It goes after a very specific adenosine receptor, the A2A receptor. And we have collaborated with a third group, a gentleman by the name of Emmanuel Planel at the Laval University here in Canada. He's a world expert on tau pathology. He developed a very simple but elegant way of testing for the effect on tau phosphorylation. I'll end on this last 2 slides here. We are a very robust effect on tau phosphorylation. This is distinct from the amyloid. There's 2 camps that are believed to be effective in Alzheimer's, tau, and amyloid. And we have an immediate effect on tau, and it appears to be a very important site on tau, which I can discuss later as we go forward. We are going to do some chronic dosing. That was an acute dosing effect. We are expecting to start some chronic dosing, so please keep an eye on our stock for that. We are clinic ready, by the way. We have completed the CGMP manufacturing. We've completed the 4-week GLP toxicology experiments to allow us to go into animals. So we expect once-a-day dosing. And again, just to remember, there is evidence that there is an acute effect, immediate effect that can be observable even in healthy people. So this is an endpoint we can follow even in the immediate part of testing our molecule for its safety effect. We might be able to have a pharmacodynamic marker that we can monitor in patients very simply. It's a questionnaire. I'll end on this last slide here. So we're trading on the OTCQB as MBCOF, on the TSX Venture as MRVL. Marvel. We're based in Calgary, Alberta. Shares outstanding, roughly 44 million. Our market cap floats between 6 and 7 million currently. Insider ownership is around 25%, so we've got skin in the game. And we would be happy to entertain any further questions here. I'll put our contact information on at the end here, and I'll tackle some questions that people have. We've discussed our academic and strategic partners. We have Fraxa, we have our group in iTours France, and we'll be announcing shortly some more partnerships and strategic partners with respect to our Alzheimer's viewpoint. Our 2020 milestones and capital needs to support. We're going to conduct our trial in Australia. We get 43% tax credits back. That's cash back. Excuse me. Happy to discuss our financing needs. Probably offline is better to do that. But our goal is to be in clinic and to try and show that our compound has a pharmacological effect even after a single dose. Can't guarantee, but that's the goal here. We have a very low cash burn. We have a very lean team here. We have about four employees associated with the company, and we've been able to achieve all this on despite COVID and a very challenging capital market here. So with respect to our shareholders, Rod Matheson, our CEO, who I will welcome to contact for further discussions if interested, was a senior investment banker and broker with a couple of large investment firms here in Canada, and established quite a good network of investors and colleagues. And so most of the stock, we believe, is in very friendly hands to us. I think we could place a very significant fraction of the stock. So we are, I think, really poised in the next year to move from a preclinical to a clinical-based company. Very exciting technology right off the bat. And we think that we can really turn the needle forward on autism. And again, we recognize that autism is very challenging, especially for a small company, but we want to unlock the value of our compound. And I think we will be a very attractive target for some kind of a business relationship with a larger pharma. I would think, there's some natural ones that you can think of that we could do. Let's see what there's another question here. Congratulations. Can you add any color to the potential partners? Yeah, in the neuroscience spaces, there's very often, if you've noticed, there's been some very significant deals being done. For example, schizophrenia, which hasn't had a real drug in the last 20 years. I mean, there was big deals being done. It's totally re-energized the sector. $14 billion deals being done. There is attention certainly going back to the neuroscience. In fact, I think if you look at VC investment, neurosciences is now second after cancer. And I would not be surprised if in the next few years, it eclipses that. Again, we've got great drugs for oncology, Keytruda, Tecentriq. All those drugs are very, very good, but neurological conditions are still underserved. I think our goal is to, be a very attractive to a takeout partner. Again, very often after phase one appears to be a sweet spot for pharma. So I think in the next 12 to 16, 18 months, I think we're going to see significant interest building us. We intend to publish our data to get it into the scientific community. A manuscript is now under review. And we intend to publish our results from all three of these studies as possible. We have some very tight shareholder base. And again, I want to stress that. And feel free to contact us if you want further information about upcoming events, financings, and so forth. We'd be happy to discuss them with you. We are very lean. We want to, as a founder, having putting my own money involved, we want to be very, very guarded of our cap table and our cap structure. So we want to be very careful about that. So, we would like to work with people who see our vision and see the potential here. I think there's very, very large potential with this compound. It appears to be very safe, and we think it hits a lot of bills. I think our drug redevelopment approach has de-risked a lot of this. We're basing our drug off an existing drug. We've made very simple modifications, logical modifications to it, and the drug is working. And we've been shown in multiple people's hands that the drug is active. So we feel very confident that this is a good asset, and I think something that's going to ultimately get the pharma attention. I think I'm basically out of time. Please feel free to contact me or my colleague, Rod Matheson. The contact information's at the board. We would be delighted to have additional conversations with you, and we thank you for your time.